Hi, I want to talk a few minutes tonight about the resources page that's associated with the general biology class. From the main BioBook index, which you can access by going to www.biobook.info, you can access your resources tab by putting your mouse over the general tab under junior biology resources, highlight resources, click on it. It will then go to this page. This is the resources page that kind of deals with general science skills in biology, and we'll come back to this throughout the year. You have a number of links on this page that are helpful to you. The screen lecture for the BioBook introduction is linked here. The syllabus of the course is linked here. If you click on this link, a PDF file will load. If your browser will not open this PDF file, you need to get to the IT department and have your browser plugin activated so that you can actually see the syllabus. In addition, lined paper, there's a link. If you just need lined paper, I print these a lot of times for tests. If you just want some lined paper, print this double-sided, you've got it, and it's got at least a page number at the bottom, 1 and 2. In addition, you have a general format sheet on writing lab reports. This link will explain how to actually make a detailed lab report. You'll do a few of these this year. I'm not sure you did any in chemistry, but you'll have a few in biology. But we'll talk more about the specifics of that later. This permission form was given to you on the first day of class. This is a replica of it. If you lose the permission form, you can always print this one and turn it in. But this lets your parents know that we will probably be walking outside at some point throughout the year, and either to look at trees on or just off campus, or stuff out in the courtyard, whatever. And this gives your parents notice and gives you permission to do that if they sign off on it. If your parents want to know what is going to come up on back to school night, what I'm going to talk about, which is right away in the beginning of the school year, this link will actually take you to the handout that I give to the parents on back to school night. So if your mom or dad misses back to school night and would like to know what I talked about in class, if you could direct them to this link and maybe even print it for them, you can show them what I would have covered in the back to school night presentation. On this resources page, you have your grades link. The grades page right now is not active, and I'm not actually in an account right now, so it won't work. But if this page was active, there would be a username and password field as well as a screen key that you'd have to type in, and when you load the page, your grades will roll out on the page. And this is one of the reasons why you need to keep your password confidential. If at any point you feel your password's been compromised, please contact me and we will change your password. Something I told you on the first day, but I need to remind you, I do not know what your passwords are. It would probably be good for me to store your passwords somehow, because I am the system administrator and I'm the teacher and I have to grade your work. But the problem is, if I store them in BioBook and it got hacked, your passwords would be compromised. And if you've used that password anywhere else, it could end up compromising some part of your online profile. So to prevent that, I only store the encrypted versions of your passwords. And so if a password needs to be changed, just let me know, and I will send you a link to a changing password form, and you'll have to put in a screen key and a new password, and it will change the password for you. This link here for science courses talks about all the courses we offer at NARINX throughout your 4 years from freshman through senior. And so you can kind of look at your possibilities, especially for senior year electives, what you might want to take, because probably you're getting interested either in science or not interested in science, and you'll want to either take more science classes or not want to take any more, and that at least that page will lay it out for you as to what your choices are. This right here lists all the biology labs, all the big ones we do. We do more than this, but these are the main ones. Clicking on any one of these links takes you to a page that is devoted to helping you complete the task of the lab, explaining what the purpose of the lab is, and how to do the lab, and then the specific procedure sheet and worksheet. So if I clicked on microscopy here, this is the microscopy lab page. Gives our basic objectives for the microscopy lab are listed here. Basic terms that are significant with microscopy are here. A potential quiz that I might give on the microscopy lab is listed there. And then there are objectives associated with, what do you need to know for a quiz when it comes to microscopy? And then a demonstration on what depth of field means. If you've lost the lab procedure for the lab, you click on the lab procedure link, and here's what I'll be handing you in class. So the very first part of this sheet, this will actually be printed, handed to you on paper, goes through the anatomy of the microscope, the procedure, the terminology that's going to be important to the procedure, and then special notes that are associated with this procedure about being careful with the microscope. And then, part A, part B, part C, part D, and part E, what we're going to do in the actual lab. And then there's a worksheet associated. It asks you to write down your observations, and then there's questions that you need to answer. And you'll be turning that part in and get it and have it graded. So the lab sheets are there to help you complete the labs and understand what we're doing in the labs. Now, there's a important thing about biology study skills. Please don't wait till the night before a test to study for it because biology is a challenging topic, and there's a lot to know for each unit, and it is very difficult to cram everything in the night before. You want to read your outlines as early as you can when studying a chapter. You want to know the answers to your homework when you walk into the test. And you're going to know that, and you want to do that by actually studying them when you do the homework. If you simply copy things from BioBook into your notebook and don't actually try to learn them, you're going to be in trouble when you get to the test because you won't know the answers to the big questions, and those are going to be on the test. So you definitely want to study your homework answers when you're doing your homework. You want to make flashcards or use the flashcards that are available to you on your study sheets, which will help you study. You definitely want to study every night, at least a little bit, in every one of your classes, including biology. Even though we have class every other day, the idea is that you would spend at least 20 or 30 minutes per night on each class to study. And then you want to use your study sheet and the links associated with the study sheet to bounce around and look at the information provided on each unit. Definitely make drawings in your notebook to map out relationships and the associations between parts, and definitely see me if you need help and I can help you out. Now, some of the basic science skills that you're expected to master by junior year in high school are conversions. You can't get away from conversions in life. You already know how to do a lot of conversions. You can figure out how many, pieces of candy you can buy during Shout Week at lunch based on how much food you have. If each piece of candy is 10 cents and you have $1, you can quickly figure out in your head that you're going to be able to buy 10 pieces of candy. Well, in science, we often have to convert units. For example, inches to feet, or feet to kilometers, or pounds to kilometers. Almost all branches, almost any career choice that you make, you're going to be converting something. If you're in nursing, you'll do all kinds of conversions to figure out dosages for patients that are under your care. So at any rate, this first chapter in the introduction, you're going to be dealing with the metric system. You definitely will need to memorize what kilo, centi, milli, and micro mean. And you're going to need to be able to make conversions between different units. So, for example, if you've ran 5 miles, how many kilometers is that? Or if you measured 5 teaspoons of NyQuil, how many milliliters of NyQuil are you taking? I would recommend you use the conversion factor method. For example, in this first problem, number 1 here, it says convert 15 inches to feet. Notice what we're doing here. You take 15 inches, you write that first, then you write the multiplication symbol, and then you write a fraction bar, that's the line here, and then you write a conversion between inches and feet. And inches goes on the bottom so you can cancel it, and feet goes on the top so that you'll convert it. And 1 foot equals 12 inches, that's the ratio. So you end up taking 15 times 1/12, which mathematically is 15/12, which is simplified to 1.25 feet. Never, never, ever, Taylor Swift ever, leave your answers as fractions. Always put them in a decimal form. Science is about approximation. It is not about abstract numbers. So if your answer is 7/6 feet, you do not report the answer as 7/6 feet. It's 1.25 0.18 or whatever 76 actually divides out as rounded. You want to round appropriately. I'll talk in class about scientific figures or scientific significance and significant figures, but you're going to need to round your answers to be appropriate to your measurements. So you'll notice the 15 inches has 2 digits, the 12 inches has 2 digits. Theoretically, this should have only had 2 digits. That should have been 1.3 feet. I don't make a big deal out of significant figures. I don't try to needle you on it, but if you report an answer with 10 decimal places because you read it off your calculator and you're starting with numbers like 15 and 12, you are going to get the answer marked wrong because you are inappropriately reporting accuracy in your answer that is simply not present in your measurements. So this answer needs to reflect the accuracy of the measurements that were taken to derive it. So we'll talk more in class about conversions, but they're coming, and you're going to have to be able to do them throughout life and on the ACT test this year. So next thing, data tables. The ACT test especially is big on having you read data tables. This is a good data table here, right? This first one. You'll notice that there's a title at the top. Each column has titles. Each row has a column. And then there are data values within cells that allow you to figure out what Fran Gothi's heart rate was after 3 minutes of exercise. These two data tables are not as good. This one's missing the title. The title should be inside of a border. If it's not, points will be taken off. A proper data table has a title at the top that's part of the data table. This title is technically not part of the data table, so that'll be a -1. This data table is sort of horrific because it has no borders. That one's going to lose most of the points it's worth. No one wants to look at a grid of numbers, and I'm not big on looking at grids of numbers, but in order to organize your numbers, you need to have them enclosed in borders or grids. So table 1's a good table. Tables 2 and 3 are not. Now, graphing. Graphing shows relationships among data. So these numbers here reflect Jane Doe's, Gertrude Jones, and Fran Gothi's performance during exercise in terms of heart rate. If you graph the data, you can actually visually see which student would be in better shape, physical shape. We look at graphs all the time in business. Those are stock market graphs or business expense or income graphs, and they quickly tell people whether businesses are making or losing money. You need to know the basic components of a graph. Every graph, also like a data table, has a title. There are labels, just like data tables have labels on the rows and labels on the columns. Graphs have labels and units on the y-axis and labels and units on the x-axis. Notice the graph has labels and units, heart rate in beats per minute, exercise in minutes. The graph is the same way. It has to display units. And then there's a graphical relationship. You can see there's a line of best fit here. That's the trend line. And then you'll notice the trend line doesn't pick up every point. We don't just connect the dots. That we don't do. The graph is a line that's among the points. It shows the average relationship among the points. And then there's an equation displayed on the graph that shows the relationship of the line. This is a bare minimum for a graph. All of these graphs are missing their slopes, but that's because they're not linear. So that's okay. There's no equation displayed because they're not linear. This first graph's good. There's a title, there's a label, there's a unit, there's a label, there's a unit. And you'll notice there's a smooth curve among the points. You'll notice this isn't a line. Why didn't we draw a line? Because the points don't indicate that there would be a line. The points indicate a gradual but exponential rise in the numbers of bacteria versus time. So instead of drawing a line, we're drawing a smooth curve. Notice that the curve does not pick up all the points. It goes among the points. This is a good graph. Why? Because the points are displayed broadly on the grid provided. The y and the x-axis are labeled, and there's a clear title, which shows the y value versus the x value. The second graph is not so good. There's a title missing. There's a point off. The y-axis is labeled with units. The x-axis is labeled without units. There's another point off. And there's an atrociously small display of the data points. This graph loses most of the points that it's worth simply because it's such a terrible display of data. This graph here is also atrocious. Somebody has decided to draw grids on the end to accommodate the points. Never, Taylor Swift, ever do this. By extending the graph outward and drawing grids by hand, you compromise the regularity of the graph plot and make this laughable. So otherwise, it has a label on the y-axis, label on the x-axis. This is a very poor title. You'd lose 1 point for the title. The title should always be y versus x. This title is just bacterial population growth. That's the y-axis, what's the x-axis? So this graph has multiple problems. This graph is horrendous because it's just connecting the dots, and it's implying that the relationship actually jumps like this, and it doesn't. The reason all the dots don't lie on the points is measurement error, and you're going to find that in science. There is no such thing as a perfect measurement system. Otherwise, the graph's okay, except for connecting the dots. Graph 5 looks okay initially, but you'll notice on the x-axis, 1, 2, 3, 4, 5 increments are achieved for making 2. 1, 2, 3, 4 jumps to 4, and 1, 2, 3 jumps to 6. Since this is irregular, this graph gets a 0 because it's actually a lie. This graph is totally invalid because there is not a consistent incremental label of the axes. Never do this to accommodate your points. You must have a fair distribution of the points on the graph. And then this last graph, time in hours, bacteria in 10 to the 6th, number of bacteria versus time. It looks okay, but we are getting very close. You'll notice this graph looks very much like this graph. The y-axis isn't labeled as well, but otherwise the graph is okay, but it will say here nearly everything is correct except the y-axis units are incomplete. The label is actually incomplete. It's not just the units. Otherwise, graphs 1 and 6 are the best in the bunch. And graphs 2, 3, 4, and 5 are horrendous for various reasons. You do need to be able to recognize what basic graphical relationships are. If you have points that are approximately on a line that are going uphill, you have a direct proportionality, and the equation will be some version of y equals kx, where k is the slope of the line. This is a linear relationship here, but it's y equals kx plus b, where b is the y-intercept. This graph is just like this graph, except that it's not going through the origin. This is linear, but then again, it's not a direct proportionality, and that's because it doesn't go through the origin. This is an inverse proportionality where the equation is the product of x and y is a constant. If you remember last year, the Boyle's law, pressure times volume is a constant. If you graph the pressure and the volume for any gas that's contained in a chamber under various pressure systems, if you measure the volume and then graph the pressures and the volume values, you get this kind of graph. That's an inverse proportionality. We probably won't have many of those in biology. We most commonly have this and this, but we'll see mostly graphs, this first graph, the second graph, and the fourth graph. We do see exponential relationships. Life forms grow exponentially. And so you will see this fourth relationship here. The general principle for this, or the general equation, is y equals 2 to the x power, where x is an exponent of 2. And then these last two we probably won't see much, but they're rare. But circadian cycles or rhythms often reach peaks at midday and then have drops in the middle of the night. And that's where these two graphs are common, but we won't have much of that this year. So here we have a typical data table. You'll notice there's a title at the top. There are column labels showing a label in a unit, and then you have your data values that were collected in lab, and everything is enclosed in borders, which is good. And then here we have a graph. You'll notice the y-axis is labeled with units, and the x-axis is labeled with units. And there's a full title, and it shows the y versus x relationship. You'll notice the points are actually here, and there is a trend line among the points, and it happens to go through all of them. And then the equation is displayed, y equals 0.8 mils of carbon dioxide per minute times x. It's y equals kx. This is k right here, 0.8 mils. That's the slope of the line. To calculate slope, you'll have to take the difference of y values over x values. So the reason this is 0.8 is because 0.8 minus 0 is 0.8, and 1 minus 0 is 1, and 0.8 over 1 is 0.8. This is the slope of the line. So if you do that for any of these intervals, it comes out to 0.8 for the slope of the line. Here's the second one. It looks a little more confusing because the numbers are harder to work with. You'll notice all the points are not exactly on a line. But both of these graphs show a positive relationship between y data values and x data values with a positive, there's a positive slope. So the equation up here is y equals 0.07 liters of oxygen per hour per kilogram times x plus 7.0685 liters of oxygen per hour. This is the fudge factor. You'll notice it doesn't quite go through the origin. That's the 7.0. If you take this graph right down to where it hits the y-axis, the value there, right where it hits the y-axis, is 7. Where did this equation come from? Excel, the application, is really good at giving you these kind of graphs. And we'll go over how to do that, in class. Now, there will be times this year where you may have to do some sampling data. And you will see this kind of data, especially towards the end of the year. This is the resting heart rate of 10 biology students that were sampled in a class: Claire, Marissa, Maggie, Maddie, Natalie, Katie. And these are all their heart rates. This is called, in a way, a sampling data set. And in this case, it's heart rates. This right here is showing a histogram of those heart rates. A histogram is a summary of this sampling data. And this is showing in brackets heart rate in beats per minute, people 51 to 60, people 61 to 70, people 71 to 80, people 81 to 90, and people 91 to 100. So what we're saying here is there were 4 people that had a heart rate between 71 and 80 in our sampling. There were 2 people that had a heart rate between 81 and 90. Histograms are really valuable because they summarize for a population. This is a population now, group of people. It summarizes statistical data for a population. This is showing the average heart rate, and there's the error value based on standard deviation and standard error calculated by Excel. It's showing the error value. So on any given day, if we did this experiment with the same students, we should fall somewhere between about let's see, the standard error is 3.6. So 83 points, since the average is around 79.2, it should fall between about 3.6 above 79.2 and 3.6 below 79.2. The last kind of sampling we'll do in class is chi-square. Chi-square is a little confusing for students, but it basically takes a look at counting data and looks at the statistical validity of the counting data. And it starts with the premise is that it's called the null hypothesis. And this is the chi-square test. The chi-square test always starts with a null hypothesis stating that there will be no statistically significant difference between the observed data and the expected values collected in the experiment. So here's what we got. We counted red versus white flowers in a field or whatever, and we found 511. Oh, this is from a genetic study, I'm sorry. And we counted 511 offspring plants that had red flowers, and 489 had white flowered were white flowered plants. And so this came from across from 2 red flowered plants a red flowered plant and a white flowered plant, and we were trying to see whether the offspring that they made are being produced by genetic ratios. We expect, from this particular cross, we expect a 50-50 ratio between red and white offspring. The thing is, we didn't get exactly 50-50 between red and white offspring. We got 511 to 489. So it's maybe 51 to 49. And the question is that valid? I mean, so to figure it out, you have to perform a chi-square test. We expected 500-500. And by performing the chi-square test, we find out that the chi-square value is 0.484. Now, you're going to learn more about this later in the year, but since there's only a red and a white color, the degrees of freedom in this particular cross are only 1, which is always 1 less than, by definition, the degrees of freedom are always 1 less than the number of possibilities in your study. There are 2 possibilities in the study, red and white, so that means there's only 1 degree of freedom. When we look down the column here, the chi-square value is 0.484. 0.484 falls between 0.46, between 0.46 and 3.84. What that means is that we are between 50 and 5%. This is 50 and this is 5%. We're between 50 and 5% certain that these flowers were produced by the standard genetic mechanisms that we understand in heredity. And that is sufficient to conclude that the plants were produced by standard genetics. We'll talk more about chi-square later, but you will be doing statistical tests like this but we sort of walk you through in biology, especially in junior bio. We sort of walk you through the tests. We want you to be exposed to these kind of statistical tests, but we don't expect you to memorize how to do the tests. Now, we'll talk more about this in class, but when you're studying science, you're going to find that you're doing research. You can't read entire books just to find one answer. You have to search through an article, and you have to find pertinent things that you need to know, and then you need to own those. You need to read those. So when you're taking the ACT test, they expect you to use science practice. Best science practice is you basically are skimming an article, you read questions, and you quickly find answers, and then answer those questions. I'm going to encourage you to do that throughout the school year, and hopefully apply that as you're going through the tests and quizzes in biology, and hopefully by the time you get to the your testing, whether it be ACT or SAT this year, you are a pro when it comes to taking science tests. Anyway, thanks for your time. I hope this helped. Please ask questions in class if you're confused about anything in the resources.